Okay, you should be seeing my screen. Can you see my screen?
Yes.
Okay, we will solve November 2023 exam variant 43. I noticed that that variant, the third variant, is always the hardest. So I want you to solve the hardest variant so that variants 1 and 2 will be easy for you. Now let's solve this variant. Let's start. They start with the plant reproduction. Figure 1.1 is a diagram of an insect pollinated flower. Okay. Using the information, complete table 1.1. You are requested to write the name of the structure and the function. What is A? What is A? It's from the picture.
Stigma?
Yes. What is the function of stigma? Come on, that's very easy. What does it catch?
It catch
Pollen?
pollen.
Yes, of course, it catches. It is sticky, of course, to catch the pollen. These are easy questions. We need to get them done quickly. What is B?
Petal.
And what is the function of the petal?
It's colorful to attract insects.
Correct. Insects. Now F. What is F?
Sepal.
Very good. What is the function of this sepal?
Protects.
It is a pot to protect the flower.
Yes, it protects it when it's a bud before the flower opens. State the names of the two structures that form the stamen in a flowering plant. The stamen is the male or the female part of the plant?
The male part.
Stamen. So what is the male part made up of?
Stamen.
Filament and anther.
Very good. Filament and answer. When the question says name, then you have to write the name. Some students will write C and D. They will give you a wrong. This will be a wrong answer. Don't label. You can write C and D, but you have to write the name. So it is the filament and the anther. Let's look at the picture. Which is the filament and which is the anther? Which is the filament? C or D is the filament?
D.
D is the filament and C is the anther. Okay, so write the names if they ask you to write the names. Describe the stages in the reproduction of a flowering plant, from self-pollination to fertilization. Now, the first thing you will look at is the number of marks. This is 5 marks. So this means you will need to write a thorough answer for self-pollination and fertilization, describing the steps. Can someone give me a nice, thorough, organized answer? What will you start with? Give me an answer. Don't stay silent.
First, we're going to say what is self-pollination.
Correct. The first thing we will do to write a nice organized answer is that you will define what is self-pollination. Defining self-pollination itself will give you a grade. Can someone define it for me, please?
It's the transfer of pollen from a stamen to the stigma.
1 minute. From the stamen or from the anther?
From the anther.
Yes, from the anther. Okay. From the anther to what?
To
Stigma.
Okay, to the stigma of which plant? Or which flower?
to a flower in the same plant.
Very good. To the same flower or a different flower on the same
Plant.
Very good. A different flower on the same plant. So I just cannot write every detail down with this pen. Now this will give you a grade. Fine. So pollination happens. We will have the pollen that will be transferred from the anther to the stigma, whether on the same flower or a different flower on the same plant. Now, after the pollen has been transferred and it falls on the stigma, what happens to this pollen?
It forms a
Pollen
tube.
tube.
Very good. It will germinate or grow into a, you just said germinate into a pollen tube.
Tube.
Very good. And this pollen tube, where does it go?
To the ovary.
Very good. It will move down the style. How does the pollen tube keep on moving down the style? What does it release?
Release an enzyme to digest the style.
Correct. It releases enzymes that will digest the style so that it can move down the style. And this pollen tube, what is it actually carrying? What is moving down this tube here until it reaches the ovary?
The main nucleus.
Pollen.
Correct. Correct. The male nucleus. Because here they want you to discuss fertilization, so definitely you will define fertilization. So the male nucleus is moving down the pollen tube. It will go down, down, down until it reaches the.
Ovule.
What is this organ? Very good, the ovary. And what is found inside the ovary? What is this?
Ovule.
Micropyle.
Ovule. Yeah, no, the micropyle is the opening. We will mention it now. And what is inside the ovule?
Ovary.
What is waiting inside the. No, the ovary is the big organ. So the ovary has many ovules, and inside the ovules we have the female nucleus. Okay, so the male nucleus will move down the pollen tube. It will reach the ovary. Inside the ovary, we have many ovules that contain the female nucleus. How will the male nucleus fuse with the female nucleus? How do they reach? How does it enter the ovule?
For the
Fertilization.
micropillar.
Yes, don't jump to the female nucleus. It will enter through the micropyle. So the male nucleus will enter through the micropyle. Okay, and it will fuse with the female nucleus. And this is the process of fertilization. And here you can define it. Fertilization is defined as the fusion of what? To produce what?
Of the
Very good.
nucleus.
It is the fusion. It is the fusion of the male and the female nuclei in order to form a diploid. What?
Zygote.
Zygote for the seed. So, for a better definition of fertilization, you say it is the fusion of the haploid. Here mention haploid and diploid. So it is the fusion of the haploid male and the female nuclei. The female nuclei here is the ovule, and the male nuclei here is the pollen. It is different in plants than in humans. It's the fusion of the male and the female nuclei to form a diploid zygote or the seed. So here you gave all the points that they need in order to score a 5 out of a 5. You mentioned or you defined self-pollination at the beginning, and then you described the process just like they asked you, and then you defined fertilization at the end. You ended by defining fertilization and mentioned that the male nuclei and the female nuclei that are haploid, you have to mention that they are haploid, they will use to give you a diploid seed or a diploid zygote. Okay? And this is also an example of sexual fertilization in plants. Now, if you want us to look at the mark scheme, where does it go? 1 minute. Where am I? Yeah. The mark scheme will definitely show you all those details in order to take the 5 marks. Self-pollination, pollen grain is transferred from the anther. So we define self-pollination just like we did. We define self-pollination here. So this is the definition. And then the pollen grain or the tube will grow into a pollen tube. You have to mention that. And the male nucleus will move down the tube and it will move through the style. We said that the pollen tube will release enzymes. We said that the pollen tube will grow, or the pollen nucleus or the male nucleus moves through the micropyle. We said the micropyle, we said ovule, and they will fuse together. And they will correct description of haploid to diploid because you will have to define fertilization. Zygote is formed. Fertilization is described in the ovary or the ovule. So all the steps, don't miss any step, and don't tell yourself, I'm writing too much. It's always better to write more than less. Okay? So it's better to write more than write less. Now, where did it go? Where is the exam? The exam is here. Okay. Now let's continue. This is a nice question, by the way. It's a question that is out of 5 marks, yes, but it's nice because it's direct. You studied well, you will write it well. You did not study well, you will not write it well. Outline the advantages and disadvantages of self-pollination compared with cross-pollination. So this is 4 marks. So definitely they want at least 2 advantages and 2 disadvantages of self-pollination. So self-pollination, a nice and a clear answer. You write advantages, 2 dots, and you write 2 advantages. Disadvantages, 2 dots, and write at least 2 disadvantages. Give me 2 advantages of self-pollination. What are the advantages of self-pollination? I'm pollinating myself.
The screen is not shared.
Excuse me?
The screen is not shared.
Oh, I'm sorry. Where is this? Is it shared now?
No.
Still? Strange. Okay, I'll share it again from the beginning. Is it shared now?
Mm.
Is it shared?
Yes.
Okay. Tell me, what are the advantages of self-pollination? I'm pollinating myself.
It
Doesn't
doesn't
need
require a pollinator.
Very good. I do not need a pollinator. No pollinator. I can pollinate myself. What else? Another advantage.
Fast.
It is faster, of course, less energy needed. I'm pollinating myself. I want another advantage, huh? In case this question is out of 6 marks, not 4. It is faster, I less energy, no pollinators. I can pollinate in the wild, or I cannot pollinate in the wild.
Yes, I can.
Of course, this is one of the important advantages. I can pollinate in the wild in isolation. And if I have a favorable characteristic, there is a higher chance or a lower chance for this favorable characteristic to be passed on.
Higher chance.
Of course, there's a higher chance for the favorable characteristics to be passed on. It is sexual reproduction, but it is very similar to mitosis because all the babies of the offspring are very identical because of the self-pollination. Remember the table in your notes that compares between self and pollination and cross-pollination. This is important. Now, what is the disadvantage? Also, another advantage: do I waste lots of pollen or no waste of pollen?
No waste.
No waste of pollen. Okay, good. Now give me 2 disadvantages of self-pollination.
Decrease in biodiversity.
Of course, decrease in variation and biodiversity. And why is this disadvantage?
Less chance of mutations to be passed on.
Yes,
Sorry,
less
mutations to
chance
occur.
of mutation or expression of mutation, no natural selection, less chance to survive or more chance to survive if there is a change in the environment. They are more adapted or less adapted?
Less adapted.
Less adapted. There is a high chance for them to become extinct if there is a disease. This is also an easy question. Okay, they're still in plants. Figure 2.1 is a diagram of a cross-section of part of a leaf. Can someone tell me what is J?
Upper epidermis.
Correct. And what is K?
Xylem.
Correct. This is the xylem. Thank you so much. This is the palisade mesophyll. This is the spongy mesophyll. This is the upper epidermis. This is the lower epidermis. This is the waxy layer produced by the upper epidermis. Okay. And this leaf is an organ made up of the different tissues that we just labeled: the upper epidermis, the lower epidermis, the palisade mesophyll, the spongy mesophyll, and this is the vascular bundle made up of the xylem. And she said xylem because, why did you say that this is xylem, not the phloem? It's correct.
Because it's larger. I'm not sure.
Yes, correct. It has thicker walls. Okay, so this is the xylem and this is the phloem. Good, perfect. Explain and identify how the structures labeled J and K are adapted for photosynthesis. Now, what is J? We said
It's transparent, so the light can penetrate.
excellent. Again, it is sometimes can be confusing because you will tell yourself it has no chloroplast. So how does it do photosynthesis? It will help the cells below because it is transparent, just like you said, and this will make the light be The light penetrates into the deeper layers. Very good. That's the perfect answer. Now, what about K? K is the xylem. Does K do photosynthesis? Does the xylem do photosynthesis?
No.
No, but how does it help?
It carries the water and minerals to the leaf.
Very good. Its structure is to carry the water that is needed in photosynthesis, carry the water. Okay, this is called conduction, carrying of water. And how is it designed to carry the water?
The ends are broken down.
Very good. Degenerated end walls. What else?
No companion cells.
Okay, but this is not needed in the transport of water. This is flowing. Forget it. Talk to me
The
about the
walls made
structure
of
of the
the walls
xylem.
are made of lignin.
Adhesion and
You said that the walls are made up of cellulose and lignin. Okay, how is this important?
For support.
Yes, when water is moving upwards in the transpiration stream, it creates negative pressure. But still, the xylem does not collapse because it is made up of lignin, and lignin is strong and it will not make it collapse as the water is moving upwards and creating negative pressure. But there are other things in the xylem that makes it very suitable for the movement of water. It is hollow, no end walls, no nucleus, no cytoplasm, no organelles. It's a dead tube. No obstruction of water. So discuss the structures of the what? Of the xylem. Okay, it has lateral pits or openings for the lateral movements of water. You studied xylem in detail. Now let's not waste time. An experimental crust plants were grown in normal atmospheric and high carbon dioxide concentration. So I would underline that because this would be the independent variable. The transpiration rate, the mean number of chloroplasts per cell, and the concentrations of starch and magnesium ions were measured, and the results are shown. So they will give you here the factor measured when there is normal CO2 and when there is high CO2. Okay? Now complete the sentences about the data shown in table 2.1. Just a quick look at the table. The transpiration rate, it is high when there is normal carbon dioxide concentration, and it is lower when there is high CO2, when there is high CO2 concentration. Okay, just 1 minute. Okay, I'm back. So, and the number of chloroplasts, when the number of chloroplasts here in normal CO2, it increases in high CO2. So obviously high CO2 sometimes increases transpiration rate. Now let's complete the sentences. Table 2.1 shows that increasing the carbon dioxide concentration caused more starch to be produced in the leaves. This shows that at a normal carbon dioxide concentration, carbon dioxide is a what for photosynthesis? Who will give me
raw material.
the raw material? Not wrong. You're not wrong, by the way. Yes, it's correct. Carbon dioxide is a raw material for photosynthesis. Can you give me another term here that can be used? Because here they're
Limiting
telling
factor.
you that limiting factor. I think both will be accepted. I have to look at the marking. 1 minute. This is which number? I would say limiting factor, but it's a raw material also. It's accepted. Correct. Good, you're thinking. So increasing the carbon dioxide concentration will create more starch, meaning that more CO2 means more photosynthesis. This shows that at a normal carbon dioxide concentration, carbon dioxide either is a limiting factor, correct, or raw material. They're both correct. Okay. During photosynthesis, dash molecules of carbon dioxide are required to make one molecule of glucose. Now how many molecules of carbon dioxide are needed to make one molecule of glucose?
6.
Correct. Okay, you should here remember the balanced chemical equation. Carbon dioxide plus water will give you glucose, C6H12O6, plus oxygen, 666. Okay, the greater quantity of starch stored in the leaves grown in a high carbon dioxide concentration means when needed, more sucrose can be produced for transport in the phloem. So the leaves, they act as a what? When they provide sucrose,
Source.
they act as a source. I move quickly so we finish this example. If anyone is confused, stop me. The greater number of chloroplasts per cell in the leaves grown and the higher carbon dioxide concentration means that more energy can be absorbed from, this is nice, I'll highlight this one. We absorb energy from where and we convert it to where? This question came in more than one exam. Energy, it is transport. This is ecology, by the way. We absorb the energy from where and we convert it to what?
From light and convert it to chemical energy.
Yes, correct. We absorb the energy from the sun or from light energy, and it is converted or transferred into chemical energy. This is one of the definitions of photosynthesis, absorbing light energy from the sun and converting it into chemical energy. And then as the plant is eaten by the primary consumer and the secondary consumer, this chemical energy is transferred along the food chain. The transcription rate is lower when the carbon dioxide concentration is higher. This means reduced loss of what from the leaves? What is lost from the leaves in transpiration?
Water?
Water.
Not water. Water is not lost from the leaves.
Water vapor.
Correct. Water vapor in a gas state. Okay, so again, I'll highlight this question for you. Water vapor. Transpiration is the evaporation of water vapor. Magnesium ion concentration is lower in these leaves because they have used the magnesium to make what?
Chlorophyll.
Chlorophyll. Easy. Okay. Very good. Until now, the exam is nice. Complete table 3.1 by writing in the percentages of carbon dioxide and oxygen in inspired air and in expired air. So you should memorize those numbers. The carbon dioxide, how much carbon dioxide in inspired air?
2.04.
0.04.
Yes, 0.04. And in expired air,
4.
4. Oxygen,
21.
21.
20 from 21. Very good. And how much goes out?
16.
16. Very good. So this brings us to the imperative question. A scientist measured the number of dust particles in inspired air and in expired air, and they found less dust particles in the air going out. Suggest a reason. Why there is less dust when you expire? Come on.
It
The
gets
hair.
stuck in the trachea?
Okay, why? What? I just gave you the answer. Now, why does it get stuck?
عشان في mucus which traps dust.
Very good. This dust is trapped by two things in your respiratory system, by hair and cilia, the hair in your nose, and cilia in your trachea, and also the mucus. Very good, correct. And this is a form of mechanical defense, by the way. Now, figure 3.1 is a diagram of the alveoli and associated blood vessels. Explain how the structure of a capillary is related to its function. This is a straightforward question also, the adaptations of a capillary.
One cell thick or
Correct.
thin, so it can for a shorter diffusion distance.
Correct. Another function.
Contain pores to make it leaky for exchange materials.
Excellent. It contains pores. And actually, when you mention that its function, you need to, they mention the word function. So mention its function. The function is for the exchange of materials. What exchange exactly?
Carbon dioxide and oxygen.
Yes, it will provide these cells with oxygen and glucose and amino acids, and it will remove the waste products, CO2, lactic acid. This will give you a grade. Okay, and give me another adaptation. Those capillaries, they are lots in number or less in number? Excuse me?
Narrow lumens.
I can't hear you, sorry.
Narrow lumens.
Yes, of course, they're very, very narrow. They're one cell thick in thickness, and they're very, very narrow. They have narrow lumen. And why is this an advantage? So the blood travels quickly or slowly?
Slowly.
Yes, the blood will move slowly, and this will give time for diffusion. Again, this is the direct question. Good. And also, don't forget that you have a very rich supply of capillaries at any capillary bed, because this increases the surface area for faster exchange of materials. Always more or greater surface area makes diffusion faster. State the name of the component of the blood that transports oxygen. Which part of the blood transports oxygen?
Hemoglobin.
Hemoglobin.
Yes, in the red blood cells. The hemoglobin and the red blood cells, correct. State the name of the blood vessel that transports blood from the heart towards the capillaries in the lungs. To the lungs. From the heart to an organ. So this is an artery or a vein?
Artery.
Correct. Now, to the lungs. What's the name of this artery?
Pulmonary artery.
Correct. Perfect. Now, state the location and the function of cartilage in the breathing system. Give me a location of the cartilage. Very obvious one.
Trachea.
Yes, the trachea is the easiest. And why is it important?
Prevents collapse.
Correct.
of
To
trachea.
prevent collapse, to keep it open, so that it doesn't close due to the negative pressure, so that you can breathe the whole time. Very, very good. Good. A student measured the rate and depth of breathing of an athlete for 30 seconds at rest, and the data are as shown. So this is his breathing rate at rest. Okay. Using the information in figure 3.2, calculate the rate of breathing. How do you calculate the rate of something?
Volume over time.
Okay, any measurement over time. Now, the measurement here will be what over time? What will you divide over time to get the rate of breathing?
One breath
Mhm.
per specific time.
One breath only in 30 seconds. And then this is not a nice question, by the way. Look at this. This is 30 seconds. And the question wants you to get the number of breaths per minute. So you have to be a little bit smart here. How many breaths did you take per second? How will you count it? They're telling you that this distance is one breath. So how many breaths did this person take in 30 seconds? Yes, let me count. This is one. This is two. This is three. This is four. This is five. This is six. Correct. So 6 breaths in 30 seconds. How many breaths in 60 seconds? In 1 minute, what will you do? Yes, we want to, how will you do it? Cross multiply. 60 times 6 divided by 30. And the answer here will be, let me do it. Do you all know how you got it? 60 times 6 divided by 30. The answer, yes, is 12 breaths per minute. Please notice the units. The units are very important. This is tricky. So this is a tricky question. Do you all know how we got it? To calculate the rate, it's the measurement that we are measuring over time. So here we will count the number of breaths over time. It is 6 breaths in 30 seconds, but they want the number of breaths in minutes. So we will cross multiply. Do you all understand it, or someone needs me to re-explain? Okay. The measurements were repeated while the athlete was running on a treadmill, and the data are shown. Now, of course, it shows clearly that the volume of the air breathed in and out increased in the rate and the depth of breathing. Using the information in figure 3.3, calculate the volume of air inspired in one breath from 25 seconds. How will you do that? Again, this is tricky. You will go first to what time? What will you do? Tell me, what would you do? I don't want to give you the answer. What will you do on the exam? You're all silent. You will tell the examiner, I don't know. What is the volume at 25 seconds? Hello? What's this? I lost connection. Okay, you're all with me. Then what will you do at 25 seconds? Give me an answer.
Doctor,
Will
there's
someone-
no screen.
There is no what?
Oh,
Screen?
خلاص, never mind. Never mind. Oh.
Okay, what will you do?
Oh, دلوقتي it's not there anymore.
Okay, now it's there. What will you do at 25 seconds to know the volume of breath? This is so easy.
We're gonna count the waves.
First, at what time? You will go straight to which time? You will count all the waves, or they're telling you they want the volume of the air inspired in one breath from 25 seconds. So you will go to which time?
25 seconds.
25.
Yes, you will go to 25. This is 20, this is 30. These are 10 boxes. So every small box is 1. So this is 21, 22, 23, 24, 25. They want the volume of one breath. So let's go to the graph. Here, it starts here. This is the beginning of the breath. And then you will breathe, 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 and it will stop here. So this is the volume. This is the volume. So the volume is from this level to this level. Now the problem is, what is this level? This is 1 and this is 2. There are 5 boxes. So every small box has a value of how much?
0.2.
Correct. So this level is 1.2. And this level is 3.2,
3.8.
4, 6, 8. This is 3.8. So the volume is 3.8 minus 1.2. What's the answer? You can see. I doubt that you will do something like this in the exam. 3.8 minus 1.2, 2.6. That's the volume. Let's look at the mark scheme in case we are making this up. Where are we? 1 minute. Yes, it's 2.6. They will give you a range from 2.5 to 2.6. So we are correct. This is the volume. Will you know how to do something like this alone on the exam? I doubt. The problem is that these questions, they are variable questions and they only come by practice. So you need to solve a lot of exams to get used to it. Okay, so this is, just read the question carefully. They want the volume in one breath, not the volume in all the 30 seconds. They only want the volume in one breath. And the volume is the length of the wave. How did I know? Because the volume is on the y-axis. Okay, so I'm going to get the volume from the base of the wave to the top of the wave. That's it. This is what I did. You all got it? And you will write it here and the unit is given. Okay, I'm going to also highlight this question so that you can go back and solve it after we're done or revision. I'll highlight it for you so you notice this question. Let's move on. Explain the effect of exercise on the rate and the depth of breathing shown in 3.2 and 3.3. Now, and again, this is a direct question. You studied the table in the lesson respiration or you didn't? How does exercise affect the rate and the depth of breathing?
It increases the rate and the depth of breathing.
Of course, the rate, it will be faster, and the depth of breathing, it will be deeper. Now, why? Explain. Why
Because
is
more
breathing
oxygen is
faster or deeper?
required for respiration.
Good. Why?
Aerobic
Because I need
respiration.
oxygen for correct aerobic respiration. Huh?
عشان contracting muscles require more oxygen.
Excellent. Because I need this aerobic respiration to produce energy because this energy is needed for muscle contraction. Again, what else? I will provide oxygen. What else do I remove?
Lactic acid and carbon dioxide need to be removed from the body.
Yes, carbon dioxide. High levels of carbon dioxide will be detected by who?
The brain.
Correct. High levels of carbon dioxide, because carbon dioxide is a waste product, and you can write the balanced equation for respiration. This will be a very added bonus. This is going to be very sophisticated. High levels of CO2 is detected by chemoreceptors in the brain, and the brain will then, it was detected by the brain, by receptors in the brain. We took all that, and then receptors in the brain will send what? They will respond by what? What does the brain do when there are high CO2 levels? And this is an example of what?
Homeostasis by negative feedback. Yeah.
Can you hear me? Can you hear me? Can you see my screen?
Yes.
Okay. Now, I was talking to myself and I thought that no one was listening. Okay. Now, here, when there are high CO2 levels, what does the brain do? Who will detect it? It's right there. Okay, it's right here. When we have high CO2 levels, who will detect it? High levels by the brain, which will send what? It is detected by the brain, by receptors in the brain, and then the brain will send
I'm
what?
not sure, but nerve impulses?
Yes, you are correct. Motor impulses along the motor nerves to the lungs, to the muscles in your lungs, which are the diaphragm and the external intercostal muscle, to increase the rate and the depth of breathing. Okay, please study the details. Please study the details, because this is out of 4 marks. You don't have to write all that, but if it can be out of 6 marks. If it's out of 6 marks, then you have to write everything. So explain. The moment you see the word explain, then write everything you remember. So this exercise, what does it do? The rate and the depth of breathing will increase. Why? Because you said it, we need oxygen for aerobic respiration to give us energy. You mentioned aerobic respiration. Write the equation. Why not? So we need this energy for the contraction of muscles. This will give you 2 out of 4. You have to mention that you also breathe faster to get rid of the excess CO2. The excess CO2 is detected by the brain receptors. The brain will respond by sending a motor impulse to the lungs. You can mention which muscles in the lungs exactly, the diaphragm and the external intercostal muscles, so that you breathe faster, so you get rid of the excess CO2. Why is the excess CO2 toxic and I have to get rid of it?
It lowers the blood pH.
Correct. It lowers the blood pH and it can denature enzymes. It's very dangerous. And I will keep on breathing faster, faster, faster until I get rid of the excess CO2 and the CO2 levels go back to normal. And this can be an example of what? When things go back to normal, to a normal set point. Very good. This is an example of homeostasis by negative feedback. So we mentioned everything. We wrote everything that can give us even more 4 marks, more than the 4 marks. Let's look at the mark scheme. Okay, where are we? So any 4 from, because it's out of 4 marks, 4 points are enough. But I advise you to write more. More is better. More oxygen is needed for aerobic respiration to release energy. We said this because I need it in exercise for the muscles,
I
for
can't
muscle
see your
contraction.
screen.
Why not? Is it now available? Can you see my screen?
No. Oh, yes,
This?
yes.
Okay, good. So we said all that. This exercise will increase carbon dioxide concentration. It will lower pH. We said it. Change in carbon dioxide or pH detected by the brain. We said it. The brain will send impulses to the diaphragm or intercostal muscles. We said it. So that you breathe faster. We said it. We will remove the carbon dioxide. And also you can refer to the adrenaline while you're exercising. It also increases your heart rate. We took it. Okay. So we said this and we said even more. We said it's an example of homeostasis. We did not miss anything because that same question can be out of 6, not out of 4. So again, it's an easy question. It's not a hard question, but most of you, 95%, will write it as incomplete. You will tell me, I need oxygen in aerobic respiration because I need energy, because I will need it in muscle contraction. And you will forget the CO2 part. You will forget that we also breathe faster to get rid of the CO2 because CO2 is toxic and the high levels are detected by the brain. And returning CO2 levels is an example of homeostasis. You will forget this part, I'm sure. This is why you will take 2 out of 4, not 4 out of 4. And that's the problem, always in paper 4. Okay, complete the sentences about the control of body temperature. The human body maintains a constant internal temperature. And this is an example of what?
Homeostasis.
Homeostasis.
Homeostasis. When the temperature moves away from the set point, the mechanism of what will return the temperature to that set point? We were just saying it. Homeostasis by
Negative feedback.
Negative feedback.
yes, negative feedback. This is the mechanism that will maintain homeostasis. Figure 4.1 is a diagram of a section of the human skin. Let's label it together. What is K?
Hair.
Correct. This is the hair shaft. And what is L?
Sweat gland.
Yes. And what is J?
Erector muscle.
Correct. This is the muscle. What is O? What is O?
Nerve.
Capillaries.
Nerve
No,
receptor.
these are the capillaries. N are the capillaries. These are the receptors, correct. They are the endings, nerve endings or receptors, because the skin has lots of receptors for pain, for touch, for temperature, etc. What is M?
Fats.
Correct. N are the capillaries and the M are fats. State the names of the structures labeled L, O, and J. We just did. L, where is L? Sweat gland. O, we said these are the receptors or nerve endings. And J, this is the erector muscle. Okay, we wrote it. Now describe how humans maintain a constant body temperature when the external temperature decreases. Use the structures labeled in figure 4.1 in your answer. How many marks? 6. 6. Now, by the way, this exam is an easy exam, even though they have several questions that are out of 5 or 6. But this exam is easy because it depends directly on how much you study. The questions are direct. They're not suggest questions. So here, you will write everything. The table that you took that maintains the homeostasis or the temperature when the body temperature drops, when it's cold. Use the structures labeled. Okay, so what will you do? How will you answer this question? You will mention every structure and how it does, or how does it work in the cold. What will you say about the J? Hmm? Are you with me or are you gone?
I'm sorry.
It's
Is it
contract.
Yes?
is it cold or hot weather?
It's cold. It's cold. They said it's cold.
The muscle will contract. The erector
Correct.
will
So
contract.
J is the erector muscle. So how will you answer this question? They want you to refer. So this is how you will answer it. I want to show you. You will go to this question and you will write every single letter and its name and what will it do so that your body temperature keeps warm in the cold temperature to keep you warm. So J, for example, J, two dots, J is the erector pili muscle. What does it do? It will contract, like you said. Why does it contract?
So
To
the
prevent
hair
further heat loss?
So the hair will stand up or lie down flat.
stand up.
Very good. This hair will stand up. And this hair is also labeled. The hair here is K. So the hair or the K, because they said refer, will stand up. And when it stands up, why is this an advantage?
Traps a layer of hair.
Correct. It will trap a layer of insulating air. Good. I'm finished. You know, if I am you, what will I do? I'll go to every letter and I will cross it out. For example, if I talked about J, so now I talked about J and K. I'm done. I mentioned J and I mentioned K. Now let's talk about L. What is L?
Sweat gland.
Sweat
Very
gland.
good. So the L, these are the sweat glands. And what do they do in the cold? Do they produce sweat or no sweat?
No sweat.
No sweat. Finished. So I mentioned it. I'll take a grade for that. So I'll cross it out. Let's go to M. How does the M help you in the cold? What is it anyway?
A fat layer.
Correct. And the fat layer, do they help you in the cold?
Yes.
Insulation.
Yes.
Layer of
They provide insulation. Very good. They keep you warm. So I finished M. I'll cross M out. What about N? The most important, actually. What is N?
arteriole.
Correct. These are the capillaries or the arterioles. And then what does it do? These arterioles in the cold.
Constrict and undergo vasoconstriction.
Correct. Correct. Be sure of your answer. And why does it constrict? So there is more blood flow that will reach the capillaries or less blood flow will reach the capillaries.
More.
When it constricts, it will become narrower or wider.
Narrow.
Narrow.
Wide.
Yes, it will not be able to go to the capillaries and it will enter the shunt vessels instead. And it will move away from the surface of the skin or closer to the surface of the skin.
Closer.
Habibti, how close? This is my arteriole. And here is the capillary. This is the skin. The arteriole will constrict. It will become narrow. So less blood will reach the capillary and more blood will enter the shunt vessels, remember?
Yes.
So less blood will enter the capillaries and more blood will enter the shunt vessels. So when the arterioles undergo vasoconstriction, less blood will reach the capillaries or less blood will reach the surface of the skin and less heat will be lost by radiation because I'm cold. I don't want to lose heat by radiation. More blood will enter shunt vessels and the blood will remain in the deeper layers of the skin, not close to the skin. Okay? So here I talked about N, finished. Now what will I say about O? What is O?
To detect the heat change.
Of course, you are correct. So all these are the receptors that will detect the cold temperature and send impulses to who?
The spinal cord or the central
To
nervous
the
system.
brain. Yes, you are correct. To the central nervous system, perfect, because it's involuntary. To the brain or spinal cord. And the brain will respond by sending a message to the arteriole to do what we just did. Are you with me? Did you understand that? So this, again, is a straightforward question. It's not difficult at all. It's very easy. If you studied this part well, you will answer it very, very well. And how do you give a perfect answer? You will discuss every label separately, so you don't miss anything, so that you take the 6 marks. You have here, actually, they're nice. They gave you a picture. They can ask you this question without a picture. So you have to remember everything on your own. We said there was one way to remember all the points. Remember it? What was it?
C.
Correct. C. And C to the power of 3, S to the power of 3. What was the S? The 3 S's. Who will remind me?
Sweating and skeletal
Sweating.
muscles.
Correct. We did not mention the skeletal muscles, but we can. It's not in the picture. But the skeletal muscles, do they contract or they relax?
Contract.
Yes, because I shiver, and shivering makes me warm. And what was the last S? So we said sweating, shivering,
Shunt
and
vessel.
what was the last? The shunt vessels. We mentioned it here. E was the erector pili muscles, and A are the arterioles. We mentioned all of them, and in addition, we mentioned other things because they are in the picture. So you'll go to the picture because they said clearly, refer to the picture. So we will refer. This is how many labels? 1, 2, 3, 4, 5, 6. That is why this question is out of 6 marks. Because you will talk about everything in the skin, everything in the figure, and how does it help you when you're cold. That is why it's out of 6 marks. Okay? Now, again, 95% of you will write what they remember. You will not read the question carefully, and they will not refer. So again, you lose a mark or 2 out of 6. This is your problem, and this is resolved when you practice more. And I can notice that you improved because you are practicing paper 4 and you already improved. So I hope this question is now understood. We know it very well, but we want to learn how to answer a question like that. When they tell you refer to the diagram, then you refer to every label in the diagram. And we notice that we have 6 labels, and that is why the question is out of 6 marks. So use the structures labeled in your answer clearly. So every time you will go use a label, you will discuss, and you will get the 6 out of the 6. Clear? Okay, the blood glucose concentration is maintained at a constant set point using the hormones glucagon and insulin. Let me erase that. State the organ that releases glucagon. What releases glucagon?
Pancreas.
The organ, yes, the pancreas. So I'll write pancreas here. Describe the effect of glucagon, not insulin, glucagon. What does glucagon do?
Increases glucose levels in the blood.
Correct. How does it increase the glucose levels?
It sends to the liver so it can convert glycogen into glucose.
Correct. Perfect. Glycogen is converted into glucose. Now, one person only is participating. Is everyone with us here joining? I want others to speak up. State 2 cell structures found in both the animal and the plant cells and bacterial cells. Common, something that's common.
Cell wall and nucleus.
Habibti, animal cells, they have a cell wall,
Sorry, I meant cell membrane.
and the bacteria, they have a nucleus.
Ribosomes.
Yes, correct. Thank you so much. Ribosomes, cytoplasm, ribosomes. Okay. State why bacteria are useful in biotechnology. Again, direct question. Very easy exam. Why bacteria is used in biotechnology? Come on.
Making of insulin.
Yeah, but why do we use bacteria? Why specifically do we use bacteria? Bacteria they have some features.
Reproduce rapidly.
Correct. Rapid
Lack of
reproduction. Something
ethical
else.
concerns.
Good. Rapid reproduction. No ethical concerns over their use. I want a third reason. At least 2 more.
They have the universal DNA code.
Yes. They have a universal code. I have a male student here. Speak up. You wanted to say something. What also do they have that I use as a vector?
Plasmids.
Correct. Okay. They're cheap, they're available, they're easy to multiply. No one is concerned. Genetically modified bacteria were grown in a fermenter. The number of bacteria was measured and the data are shown. Now, this is the popular graph that we took in 2 lessons in ecology and I think in genetic engineering. So the number of bacteria here in millions per centimeter cubed and the time. On figure 5.1, draw an X to identify the lag phase. So where is the lag phase? The beginning, A, B, or C here? A or B or C? Where is the lag phase?
A.
Yes, A is the lag phase. Okay, now what else? On figure 5.1, draw a Y to show where the birth rate is equal to the death rate. It's A or B or C. When the birth rate is equal to the death rate, come on.
C.
Yes, here is the Y, the C, when the birth rate is the constant phase. Okay, calculate how long it takes the number of bacteria to reduce by half after the bacteria has been in the fermenter for 24 hours. Okay, what do you do? Which time do you go to? They said they want to know the population of bacteria and how long it takes to be reduced by half after it has been in the fermenter for 24 hours. So what do you do?
We go to the 24-hour mark.
Yes, you do. This is a 5-hour value. Okay, so this is 21, 22, 23, 24. 24, I'll move up, 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 up. The value is 10. It was 10 million bacteria. What is the time needed for this 10 million bacteria to drop to 5?
We draw a line across.
Yes, and let's see from 24, this is 36. So 36 minus 24. What's the answer? Let me get the calculator. 36 minus 24, 12. 12 hours and it will drop from 10 to 5. Did you all get it? Did you all see how we got it? Let's see if our answer is right or wrong. Yes, our answer is right. Okay, I hope all you all know what we did. We went to 24 and the number of bacteria was 10 million. And then we wanted to see the time needed for the number of 10 to drop to 5, which is half. I'll go to 5, it will be 36. So 36 minus 24. It took me 12 hours for the number of bacteria to drop. I hope you all know what we did. Describe and explain the change in the bacterial population size from 24 to 50 hours shown in the figure. How many marks? 3 marks. Who will answer this question? This is also very easy. What happened to the bacterial population from 24 hours from here until here? What is this phase?
Death phase?
Of course, it's the death phase. Of course. And what happens in the death phase and why? Again, it's a direct question.
There was lack of food. There was a high level of toxins.
Let's just mention what
Overpopulation.
happens in the death phase. Correct. Everything you said is correct. The death phase here. So there are more deaths than births. The bacteria will die. Why? This is the explaining part. All that you said. There is less food. There is a lot of limiting factors. Use the term limiting factors. They like it. There is a lot of competition. There is no space. There is accumulation of toxic waste products. There is lack of oxygen. It became very acidic. Anything. All what you're saying is correct. The fermenter is kept at an optimum temperature for the bacteria. Explain why this is important for enzyme function. Again, this is a very direct question. Why is optimum bacteria important for enzymes? Very easy. When we discuss temperature, we always discuss what optimum temperature means. Optimum what is provided for the molecules to move.
Activity of enzyme.
Yes, but always when temperature is involved, we always mention which type of energy.
Kinetic energy.
Correct. So it is optimum kinetic energy. The optimum kinetic energy that will provide the most efficient or successful what?
Collisions.
Correct. The successful collisions. So that I have maximum or optimum reaction rate and maximum production of what? What's the keyword that we always use here with enzymes?
Enzyme substrate complex.
Excellent. Enzyme substrate complex and maximum production of products. Always use all the keywords here. Temperature, the moment you see temperature and enzymes in one question, always mention kinetic energy, but never mention kinetic energy when you discuss pH and enzymes. So, optimum temperature means the best kinetic energy for the fastest reaction, or the fastest or the best successful collisions, best enzyme substrate complex. Also mention enzyme substrate complex. How does it happen? I have a complementary active site that is complementary to the shape of the substrate. Write it on. Okay? Most products, the enzyme is happy, the active site is complementary, it will fit with the substrate. Write everything you know about enzymes. State that bacteria are grown in liquid that contains amino acids. Also, this is easy. Bacteria, why does it need amino acids? Can you be quick? Why does an organism need amino acids? Hello? Hello? Did I lose connection? Why does any organism need amino acids? Can you hear
For
me?
growth,
Yes, to make what?
to make proteins.
Yes, of course, to make proteins, for growth, for enzymes, for whatever. Okay, xenophytes are plants that are, the last question. They are adapted for an environment which has very little available water. What is the meaning of adaptation? What is adaptation? And this is 3 marks, so you need to be a little bit creative here. Hello? What is adaptation?
The process by which populations over many generations become increasingly suited to the environment.
Yes, you're correct. These are features or processes that will make animals or organisms adapted or suited to the environment. Okay, now I want to write another 2 sentences so that I take the 3 out of 3. You will take maybe 1 or 2 out of 3. These features, how do they move on from generation to generation? And this is an example of what?
Natural selection.
Yes. So these features, number 1, you can say that they are inherited from generation to generation because these features will make the animals or the organisms reach reproductive age. And when they do, they will pass on their genes, or their genes will be inherited from generation to generation. And here we're talking about the steps of natural selection. This is a little bit of a tricky question. Okay. The climate in the desert has very low rainfall
Miss,
and
the
very
share
high.
screen is
Again? Okay. Thank you for telling me. I'll share it again for the 10th time. Is it shared now?
yes.
Okay. The climate in the desert has very low rainfall and very high daytime temperatures. So they're giving you here a big picture of the cactus. And then describe 2 visible adaptive features shown in this figure and explain how each feature is beneficial for living in a desert. So go and point 2 things that you can see with your own eyes that will help this plant to live in the desert.
Spines.
Correct. These spines, what will you say about them?
They help them in protection to avoid being eaten by other animals in the desert.
Okay, this is right, but how is this related to surviving in the hot weather? I'm not saying it's wrong, but why don't they have broad
Increases surface area for transpiration to prevent water loss.
Exactly. It reduces surface area, so less transpiration. So this is one feature. What other feature do you see?
They're wide to hold more water.
Okay, correct. They have thick succulent stems. Thick, succulent, or thick, fleshy stems. These fleshy stems, they will store more water. Remember the picture of the aloe vera? They store water, correct. So these are the 2 things that are visible. You can see them clearly. Table 6.1 shows some data about stomatal density in the leaves of one plant that is not an xerophyte and 3 xerophyte plants. Using the information in Table 6.1, estimate the total number of stomata in an ice plant leaf with a lower leaf surface area of 8 centimeters squared. This is the ice plant. It is an xerophyte. The upper leaf has 0 stomata, but the lower leaf has 42 stomata per millimeter squared. This is a very tricky question. How will you know the number of stomata in 8 centimeters squared?
Cross multiplication.
Yes, but there's something tricky here. So you have 42 in
Multiply by 10 squared.
1 millimeter. Okay. This is in 1 millimeter squared. How much in 8 centimeters squared? To convert 1 millimeter to 1 centimeter. Sorry, to convert 1 centimeter to 1 millimeter, what do we do? We multiply by
10.
multiply by 10. But this was centimeter squared. So you're doing it 2 times. You multiply by 10 and then multiply by 10 again. So first you will convert the centimeter to millimeter. So 8 times 10 times 10 to convert it to millimeter squared. So it will be 800. So you will cross multiply 42 times 800. 800 times 42 divided by 1. The answer will be, let me see, 800 times 42. The answer will be 33600. Let me see the mark scheme. Just give me a second. Yes. 33600. 8 centimeters squared. You will have to multiply it by 10 2 times to get it into, or to convert it to 800 millimeters squared. And then this 800 you times 42. Now, I doubt that you will notice that. This is so tricky. And we are all not good in math. So please highlight this question and look at it again. Did you all understand what we did? Because it is squared, so we multiplied by 10 and then multiplied by 10 to convert the 8 centimeters squared to millimeters squared. Did you all notice that? Or did you all understand it now? No.
Yes.
Okay. Now the last question. Hopefully, we have another question. Oh my God. The exam is so long. Explain the data shown in table 6.1. So go to the data and just explain it and give numbers. In the xerophytes, why do they have less stomata in the upper surface and more stomata in the lower surface? I think it's obvious that there is always more stomata in the lower surface of the leaf than the upper surface. Why? Give any two reasons. Give me an answer. Did I lose you? Hello? No, I did not lose you.
Water loss.
Yes, to prevent water loss. First, you will write some data. You will say that I have more stomata in the lower or the upper surface?
In the lower surface.
In the lower surface. Now, give me two reasons why. The stomata is hiding from what?
Sunlight?
Yes, yes, yes. I want you to be sure. It's not the stomata is hiding from the sun. The stomata is hiding from what else?
Low water potential.
Okay,
Because
how do you
water
write that?
moves
In what context? Excuse me?
because water moves from high water potential to low water potential.
Yes, correct. But why, if it is on the upper surface, it will move more? So I want to reduce transpiration. I think we all agree. So the stomata is hiding from the sun, and it is hiding from what is on the upper surface?
Humidity.
Humidity is actually more humidity will reduce transpiration. From the wind, from the sun, from the heat. Okay, so when the stomata, this is the leaf, when the stomata is on the lower side only, it is hiding from the temperature, it is hiding from the sun, it is hiding from the wind, it is in the shade, because I want to reduce water loss. But still, the stomata is important for gas exchange, but I don't want to expose it so that I reduce water loss as much as possible. It's an easy question, you will get it most of the time. Okay, the last question. There are xerophytic forests which are threatened by human overexploitation. Suggest reasons why is it important to conserve xerophytic ecosystems. This is the only ecology question in this exam. Why is conservation important? And this question is very common. Why do we conserve our ecology? Give at least two reasons.
To prevent extinction?
Extinction.
Yes, we want to prevent the reduction in biodiversity. So not to reduce biodiversity. What else? Give me another reason. Give me another reason. Why am I conserving my plant? I'm not cutting it down. Come on.
I'm not sure, but so we don't disrupt food chains.
Yes, you are right, Habibti. I swear to God, you're right. So that I don't destroy food chains. Plants, this is food for animals. So not to destroy food chains. So that the plants do not become extinct. So that I do not make my plants reduce plant extinction. Anything to protect vulnerable ecosystems. You have a list in your notes. Go study because this question is common. To protect vulnerable ecosystems because these plants are used for food and they can be used for medication. Okay? For any reason. So these plants can be used for a lot of things. They can be stored seed banks. I need to conserve them. I have to protect them. Okay, in my opinion, this exam is not hard at all. On the contrary, this exam is long and all the questions are straightforward. It depends directly on how well you studied. And this exam is important because it includes a lot of lessons. It included plants, reproduction, and transport and plants. And there was no inheritance here. And it included the microbiology or genetic engineering and the graph, the lab phase. and it included homeostasis. Homeostasis is not very common in a lot of exams, but here it was detailed. Like if you did not study this lesson, you will lose at least 9 marks, 6 marks here and 3 marks here. Okay, this question was tricky. I highlighted it for you. So this exam is an easy exam, but it's a long one. Now, if you noticed, when they get you a lot of direct questions, the exam is longer. When they get you a shorter exam, it is usually a lot of suggest and tricky questions. So I highly advise you to resolve this exam, because this exam has a lot of tricks and it makes you revise a lot of lessons. And I want you to understand how to solve a question like this, in which they tell you clearly, use the labels and discuss it. So we will discuss every label separately. Okay? But I also will tell you something. I noticed an improvement in your answers because you are practicing and you already did improve. Anyone has any questions before I go? Hello?
No, thank you.
Thank you.
Okay. Again, while you're solving, if you see any question, send me the question and I'll answer you immediately. Best of luck. Bye-bye.
Thank you. Bye.
You're welcome. Bye.